I'm going to talk today about AMR genomics from reads to reports. And as I said, so I'm I guess technically Dr. Finlay Maguire. I'm an assistant professor at Dalhousie University in computer science and epidemiology. I have affiliation with Sunnybrook Research Institute in Toronto for pathogen genomics work, as well as various involvements in public health aspects of AMR and COVID. So today I'm just going to give, and this should be like a pretty quick, and I'm hopeful I can do a bit of a Q&A. I can do a bit of quick Q&A afterwards to talk about some of those specific questions that come up. So first, I'm going to talk quickly about quality control from your sequencing data. So how do you know whether your sequencing data is any good before you try and waste all your time in analyzing it? Briefly on trimming and error correction of that data. How you then go about assembling a genome, like what commands you can use, and this is only going to be one example. And then, the nuts and bolts of using AMR Finder, as Dr. Feldgarden's talk kind of explained all the basics of and did a great introduction to it. Standardization of outputs using harmonization. And then I'm going to talk a bit about like, okay, if you're doing, this is how you do it for one sample. What about 10,000 samples? How do you change this to do work like that? The things that won't be covered in this talk is read-based analyses. So we're going to skip over the trials and tribulations of analyzing the reads directly that being touched on a bit of cases. We're not going to talk about metagenomics because, again, that's a whole other bag of fish. And I'm not going to talk about the many alternative tools for a lot of these steps. There's as many read trimmers and genome assemblers and AMR gene prediction tools as there are days of the week. And so I'm not going to talk, I'm just going to choose one of each, and we're going to talk about that. But there are many out there, and it is worth evaluating more than one for your particular problem. So the kind of main thing today is, as I said before the break, is we have a sort of I put together a quick sort of GitHub repository that does contain both all of the commands run today, examples, some little test data that you can find at github.com/fmaguire/AMR training workshop practical. And then each of these folders just contains kind of each of the steps I'm going to walk through, as well as an implementation of a simple kind of workflow to rerun everything. And then there's a fairly long README that kind of pretty much goes through what I'm going to talk about today, just in more detail and more specifics. So first things first, okay, you've got some reads. You want to do some AMR detection with the AMR genomics. So the first thing to do is set yourself up for success. Unfortunately, for the user-friendly side, bioinformatics does rely heavily on the Unix shell. This is sometimes called a terminal. You would access this through a thing called a terminal. And the easiest ways to get access to a Unix shell is either be running Linux, running Mac OS X, so you'd open the terminal app in that, and then you would have access to a shell. And you can also do it through Windows now, thanks to the introduction of Windows Subsystem Linux 1 or 2. So this is so you can run your Windows, you can use this part to run kind of little Unix shell that will let you run and do a lot of these bioinformatics things. There's some probably rough edges a little bit with Windows Subsystem Linux 2, but that's the kind of, there are lots of different ways you can get access to Unix shell. And while yes, it is not as nice and friendly as a point and click thing, a lot of bioinformatics is built for us and kind of assumes you kind of know how to do it. There are materials to like learn the basics of this, especially software carpentry materials, linked in that repository. So if you want to learn more about like how you open, how you navigate a shell, how you deal with all that, jump in there. And then as Dr. Feldgarden mentioned, actually installing bioinformatics tools, you got your shell, how do I install these bioinformatics tools? Well, package managers and containers make installation a lot easier. So again, there's a lot of different package managers, like one that you might be familiar with would be something like the App Store on your phone or something. That is a kind of package manager. And containers are just a way of kind of trying to hide it from a lot of the mess of your computer and kind of package it all up in one go, like a bit like a virtual machine if you encountered them. So containers and environments are sometimes separate from package managers and containers for installation, because they just stop you getting tools in the way. So if you install this tool, but that one interferes with this other tool that you've also installed, an environment lets you install them separately. There's a tool called Conda, in particular the BioConda channels that you can set up on it, that really provide both of those features, both an ability to easily install tools and an ability to easily install tools and deal with the deal with bioinformatics tools, especially via Bioconda. So there's, again, instructions on the repo of how you install Conda on your system, depending on your system, it's a full set of instructions, and then instructions on the Bioconda website on how you set up Bioconda, how you set up Conda to work with bioinformatics tools. What this means is once you've done that, is you have really this nice feature where you can just type Conda create, name an environment, and then just name the bioinformatics tools you want to install. And then then it'll just run through that. It'll install all those things for you without much need of complexity or difficulties. And then you can just open that environment with Conda activate AMR. And again, if it's going too fast, there's kind of deal with it. You can touch it at your own pace in the README. So the first kind of key major point of AMR genomics is garbage in, garbage out. If you put in bad data, you're going to get bad results no matter what you do. And the key kind of aspects of that are quality control. So I would say one thing they'll always consider, especially when setting up a new process or something, is the inclusion of positive and negative controls. So positive control is just you want one of your samples to have a known either AMR gene in it, plasmid, genome, phage, whatever, just a known sequence. Then you can run through your whole workflow, and if you still get that sequence you expect to come out of that positive control, you're good. It's a good sign that your things might work. If you get something else, something's wrong, you need to kind of investigate that. Similarly, a negative control, and I know this is familiar to anyone that's done a lot of lab work, but for AMR genomics, that could just be one of your samples, just water, doesn't contain any DNA. And if you get DNA samples coming out of that, you get AMR genes being predicted out of that, again, something's gone wrong. There's been some contamination or something very strange is happening. And then that basically lets you kind of contextualize, like, can I actually trust my AMR, my actual research sample results and AMR predictions when they come out from my actual research samples? The other thing to really do quality control wise, and Inês mentioned this, is contamination checks. So there's a bunch of tools that let you basically try and get a taxonomic profile of your samples from the reads themselves. And these are often used in metagenomics, but it's highly recommended you would use them for genomics because it basically gives you an idea of what contamination is turned up. Like, for example, if I'm sequencing an E. coli here and I run something like Kraken, then make a plot with another tool called Krona, and I see most of the reads I'm getting for seem to be eukaryotic, especially, and not even like eukaryotic we necessarily expect, like a lot of random microbial eukaryotes rather than human, which you might expect from a clinical sample, we might want to reinvestigate this or deal with some ways to try and clean our data. So contamination is a really important thing to check for, especially when you're making calls about AMR genes. And then the other things are just kind of your general sequencing quality checks. So, quality scores, overrepresentation of certain sequences, number of ends that turn up in your reads. And one way to do that is usually integrated into a tool that does the trimming of your reads. So that's a tool called FastP that I quite like. And that does that kind of sequencing quality control, makes reports for you, as well as removing both low quality bits of sequencing data and getting rid of things like adapters. So artificial sequences you've introduced to actually do the sequencing process. And so this is, again, very easy to run once you've installed it with Conda install and Conda activate. And it's just FastP, and then you have your input sample for the two sets of paired end reads. So this is for short read Illumina data where you have two sets of reads for each fragment of DNA you sequenced. And so you just put them both in as input, and then you want your outputs for the name to your trimmed reads, and you hit go, and it both trims it and then generates a really nice report that you can open in a browser. It contains all that information about what's going on with the trimming and the, how many what percentage of your reads were good enough quality to keep in, how many contained lots of ends, what adapters got removed, etc. So there's a really nice report where you can see, you can both look at the quality control, you can look at before and after, and you can see whether the trimming actually seems to have made a big difference. Just a quick note that the reads in the repository are simulated to be quite simple, so trimming isn't going to do very much to them. Okay, so we have we've done our nice robust quality control experiment. We have our trimmed reads. How do we turn reads into a genome? And again, there are many, many, many ways to do this. You can map it to a known, if you have a reference genome, you can just map those reads to that genome and assemble from there. But today we're going to talk about de novo assembly mostly. So this is where we take our clean reads and we kind of assemble that. We stick them all together and to get long chunks. Normally we don't get one nice long clean contig for our entire genome, but we get some decent length chunks if everything else has gone correctly. So how does this work? And again, as is the theme today, this is a topic of this can be a topic of an entire extended workshop all by itself. In fact, is in the Canadian Bioinformatics Workshop on a set genome assembly. But essentially, the very rough idea, and this is one particular paradigm. Another paradigm is more common called the Browing graph assembly, but they're approximately the same idea. There's just some quirks and differences in the way they actually work. Essentially, what we do is we take all our reads, we find kind of by either splitting everything up into k-mers and comparing them, or just looking directly at the reads, we look at how they overlap with one another. And then we can use that to kind of make a graph that has all the connections between these reads. This is an assembly graph, you might have heard that term. And then we kind of navigate paths in that graph to pull out those contigs. However, there is quite a big difference between assembly theory when you hear about it in talks like this. It tends to be this very simple picture on the left where we have a relatively simple puzzle and okay, here's the path we just put in the last puzzle piece. When the reality of genome assembly is more like this where's Wally or where's Waldo, depending on your country, picture where you have millions of pieces, lots of similar patterns. There's often missing pieces, damaged pieces, and you don't know what the underlying picture actually looks like. So this is a particular challenge. Like these many Waldos in this picture could be things like mobile genetic elements, genomic islands in particular. Those repeats and inserts, they can really make it very challenging to do assembly accurately. So one tool I recommend for assembly, and again, gone through in the practical, is something called Shovill. This is a tool written by Torsten Seaman, among the many bioinformatics tools written by Torsten Seaman, and is very, very user-friendly and does a lot of things for you in one go, which is convenient and makes quite good assemblies. So first, it kind of estimates the size of your genome from your reads, and then it kind of gets rid of any reads that would be above 100 times coverage on your genome. Why 100 times coverage? Well, there's some empirical evidence that 100 times coverage is a really good depth to recover good quality genomes. But it is also an arbitrary decision. It can do trimming optionally as well, so you don't have to run FASTP separately, but you won't get all the same quality control information. Then it does some kind of tricks to do error correction of reads using a tool called Lighter. It'll stitch together any reads that overlap with one another using a tool called Flash. And then it'll do that kind of process I just talked about, where it'll kind of look for overlaps between reads or k-mers and build a graph out of that and then find paths in that graph. And they use a tool called Spades, which is a very well-established genome assembly tool. And then the last thing it does, and a nice thing is what it does, it takes all of those reads, maps it back to your assembly, and then use a tool called Pylon to kind of try and fix any little corrections or sequence or assembly errors that are obvious from the reads. This is surprising. It can save some surprisingly difficult and messy bits. So Shovill, you just run the command line. Again, very simple. You feed in your trimmed reads or your raw reads and provide an output directory. That's all you need to do. So once you've run your assembly, you've run Shovill, you've got your contigs. How do we predict AMR genes from that? Well, Dr. Feldgarden told you all about AMR Finder today, and Kara told you about the problem of, Dr. Tsang told you about the problem of AMR databases. So the first thing you want to do is you want to make sure you're using the latest database because AMR genes are a moving target. So you use AMR Finder update and it will automatically update to the latest database. And then you can just run it, you just provide it with nucleotide, the name of your contigs file, the path to your contigs file, and then output file and output that TSV that Dr. Feldgarden went through in detail. So again, really simple to, like once you're on the command line, the stuff's all pretty simple to run. And then next, back onto Inês Mendes' talk. Okay, you've got your AMR genes, you've got your assembly, you've got your reads, you've gone through those 3 steps. How do we standardize the AMR gene detection? So how do we compare, say, if we want to try a few different AMR tools? And the problem is AMR Finder Plus, for example, has contig ID, gene symbol, percentage coverage, or reference sequence. On the other hand, ResFinder, has the same information often and a lot of overlapping information, but in ResFinder it's called contig name, gene, coverage. Similarly with RGI, contig, best ARO, percentage length of reference sequence. So what you can use is you can use harmonization and again, Inês went through this in a lot more detail, to basically translate all of these different fields and different tools to one standardized set of names. And this is a little bit, there's a little nuances in running it, but essentially it's again, a simple command line tool. So you just harmonize the name of the tool you're wanting to run. And then there's a couple of bits of metadata that you need to provide extra. So not all tools will contain all of the metadata that's really important to compare across tools inside its own output. So for example, for AMR Finder Plus, it's quite useful just to add in what is the version of AMR Finder Plus you're using, what is the version of the database you're using, and what is the input file name. There is an option to add this in AMR Finder Plus, the file name or the sample name, but lots of people don't use it or don't see that the options. And then once you've got your then that will spit out a harmonized AMR report. Okay, great. You've got your harmonized report. It's all in a consistent format. Well, then you need to give it, you want something useful to happen with this data. So who'd you give it to? How do you summarize this? Do you make it into graphs and put it in a paper? Do you give it to your clinical colleagues so they can make treatment decisions? So one of the nice things is harmonize, summarize, as well as RGI and RGI heat map, and also any of the sets of many tools out there for making graphs and looking at data. Is you can kind of use them to either create these kind of interactive reports, such as in harmonize, summarize, and that's just using the interactive summary, chucking in the reports, and all the reports from all the tools you've run, and spit out a summary. Or you can just even read in those big tables, spit out as a big TSV, big table, read it into your favorite plotting library or data tool. Like even Excel is fantastic for doing this kind of data exploration and summary. This is the bit where there's almost an infinite range of ways you can summarize the data. And this is where really talking to the people who are going to use the data comes in very, very handy. Okay, so I've got a bit of time left. So that's great. That's how we do it for one sample. But what about tens to thousands of samples? So our option one is we go through these commands, and these are just a simplified version of those commands I just showed you, just with the command, the input, and the output, and say, okay, this is sample one. So this is what we just run. So we have our sample one. We could type in all of these commands for sample one, and then we go back and type in all of the commands for sample two, back again, all of the commands for sample three. But this is a lot of typing. Humans make mistakes. The more typing you do, the more likely you are to make mistakes. It's easy to miss when things don't complete, or times when you've accidentally run it on sample two twice and you've not run it on sample three, etc. So I don't recommend doing like this. If you've got lots of samples, there are better ways. So one of those ways, and one of the kind of ways that you're first taught, is a bash loop. So instead of having all of these commands written, whatever, is we can make a little loop using bash in this case, or any programming language, that basically just runs over all of the samples and just runs these commands using each of those samples. So this is exactly the same. So it just means for each sample, so 1, 2, 3, 4, 5, 6, all the way up to 10,000, run this retrimming, assembly, AMR gene detection, and harmonization steps. And it just will run through all that. And that's great. That will solve a lot of people's use cases. But it's easy to miss if something goes wrong. Because if it's running 10,000 things, if something goes wrong in the middle, it's very hard to keep track of that. And how, sample 900 failed. But if you weren't paying attention, you're not going to miss it in a simple implementation like this. The other challenge is also this could be quite inefficient. This is running it in serial. So it's running sample one and doing all the steps, running sample two and doing all the steps, which if you have a more powerful computer, or you have multiple processors on your computer, can be quite inefficient. You're not using your computer that well. So this is where option 3 comes in. And these are something called workflows. And there's many different workflows, tools, and managers and implementations of individual workflows, but I'll go through that in a second. But essentially the idea here is what you're doing is you're taking very much that inner bit of that for loop and converting it into just some degree of specifying the rule. So instead of, saying fastp, raw read, sample, trimmed reads, whatever, we can take, AMR Finder Plus. If we want to run AMR Finder Plus on a whole bunch of sequences, we can, in this case, using the example of a tool called a workflow manager called SnakeMake, we can write this tool that says, okay, this input, so as input, it takes the assembly, it takes the contigs, and it'll have the sample name at the end, an ID. And I wanted to output the AMR predictions and put an ID at the end of that. And then to actually to go from input to output, it needs to run this command. So AMR finder, nucleotide input. So it will replace that with a particular set of contigs, and output, and it'll replace that with this particular output. And what you do is you basically write one of them for each of the steps. And then it can basically, you give it to the workflow manager. It's now its problem. And what it'll do is say we have 5 samples, it'll kind of go through and be like, okay, to get all of these outputs that we wanted, I'm going to have to run Shovill for each of these 5 samples. Then I'm going to have to run AMR Finder Plus for each 5. I'm going to have to harmonize. Then I'm going to have to kind of summarize and harmonize them all together. And the really nice thing about using workflows is, it catches when things fail for you. And say if you wanted to add new data, you can hit just run again. If you just press run again, it'll only run for new things that haven't been done. So if you've ran for 900 samples and you have 100 more, and you just add that extra 100 samples to your folder, rerun the workflow and only rebuild that missing 100. Or say one of them did fail or had you noticed there was a problem in one of the read files. You went and changed it and fixed an error in it. Again, you can just hit rerun and it's going to only rerun that one sample that's changed. So it's very kind of, it's smart. It's sometimes to be a little bit too smart for your own good, but it just saves you a lot of effort and redundancy in running things. The other thing is also it distributes over your computing resources nicely. So if you have an entire HPC cluster, workflows tools will let it kind of fire your data at that cluster and it will manage running it in parallel across everything. But even if you're running it on your own computer, it will you can fire it across your processors, give it as many cores you want, and it'll do it in parallel and use your computing resources efficiently. And there is an implementation, a very simple, basic snake-make workflow implementation for these tools that you can find in that repository. Part of the slide didn't load. Oh, that's a shame. Let's go show anyway. Why not? Pictures didn't load on this slide. Oh, well. And option 4 is the best option, the one I actually recommend. Using someone else's workflow. You can write these things yourself, but there's lots of people out there working on these workflows. There's lots of people using them. And there's lots of kind of refining and like dealing with all the problems. And like there's some really nice high quality workflows out there. So I'd say there's 3 main workflow managers that probably of interest to people here. There's Galaxy, which is the most user friendly. It's not terminal based. You can point and click on a server. And there are really nice published community workflows for that. But I would recommend make sure to check and use the highly rated ones or highly used ones, because this is not a curated set of workflows. There can be a lot of noise in there. There's SnakeMake, which is the demo I showed, which is great if you're familiar with something like Python, but the mental model is a little bit difficult. When you're specifying all those kind of rules and steps that you want to do, you kind of have to work backwards from the final output you want all the way back to your original, say, your read. So I work back from the harmonized summary, all the way back to the raw reads for each sample, which can be, it's just a little bit challenging as a mental model. The nice thing with these guys is they have SnakeMake workflows, which is this curated set of best practice workflows. So there's a nice set, usually led by the developers of SnakeMake itself, of these really high quality workflows that do a lot of checking of your data and things like that. And then finally, there's Nextflow, which I'm use quite heavily these days, which is a little bit more of an unfamiliar language. It's a little bit different as a script, like the code you have to write is a little bit different, but it has a simpler model where it works from your inputs to your output rather than backwards. And yeah, this is probably the clearest part, but the nice thing is then they have, again, this group of NF core workflows that are created best practice workflows that other people have written that are like crowdsourced commitment. So depending on your problem, I would look into those things to see if there's already a workflow that exists to do your problem. Particularly for this, there's a few kind of workflows in there, the pictures that haven't loaded for some reason. One is Torsten Siemens Null Arbor, which will go all the way from reads to generate a nice interactive report containing AMR genes, for example. And the other is Backtopia by Robert Petit at Third at, I forget where he's based. He was at CDC, Genome Tracker, and FDA, but has written a really nice Nextflow workflow that does a lot of bacterial genomics. If you're interested in mobile genetic elements as well, we've done some work in our group on something called ARETI, which is, again, is an EXO workflow, but it's really focused on detecting AMR, lateral gene transfer, horizontal gene transfer of AMR in a big set of genomes. So if you're interested in that particular problem, that might be one solution. So key takeaways. I know this is a bit of a whirlwind. Bioinformatics, unfortunately, is generally built around terminals. Things like Galaxy can help, but there's a lot of tools and a lot of things you won't be able to access via Galaxy. Highly recommend using Conda environments and containers to install tools. Always try and read the documentation and help messages for bioinformatic tools. Most tools actually do have some decent documentation that does explain a lot of the issues and use case elements. Robust quality control and controls, experimental controls, are vital, as they are in any bit of research. I would strongly consider using harmonization for your AMR gene protections, because it makes it easier to change and compare across different AMR gene detection tools. And workflows let you efficiently and robustly run an analysis over many samples. And especially, don't reinvent the wheel. Many high-quality workflows exist, and modify them if you have to. I just quickly want to acknowledge, because this is my chance to, the great speakers today, Dr. Kara Tsang, Dr. Mike Feldgarden, and soon-to-be Dr. Inês Mendes, as well as the organizing committee, myself, Dr. Andrew Page, Dr. Emma Griffiths, Professor Mark Palin, the incredibly organized Lisa Maciaretto, and Dr. Jessica Bonham, Dr. Carolyn Johnson, JPIAMR, as well as our three supporting organizations, JPIAMR, PHAGE, and MRC CLIMB-BIG-DATA. And then we have a couple of extra people who are hopefully about to join us for a panel discussion. Assistant Professor Henk de Backer at University of Georgia, and Dr. Anthony Underwood at CPGS. Okay, so let me dive into the questions, as we have 5 minutes to cover some of them.
So Finn, as you were doing your fabulous presentation, that was fantastic, and lots of people are asking for the recording so that they can watch it again and kind of absorb all of the wonderful nuggets of information that you have shared. But one of the questions that has been coming up frequently in the chat was, what about the issue of parameters? Is there like a gold standard set of parameters that are good to use in all of these different tools, or tool-specific parameters? What are your thoughts on that?
So I would say this is where short answer, no. Right. On the public health and clinical lab side, there still has not been as much accreditation and formalization of what is required for detection as there has been for other aspects of diagnostic and even diagnostic medicine. There's a lot of work happening in this area, but there's currently still no hard and fast rules. The benefit of something like harmonization is it lets you kind of compare across parameter settings quite easily, and even across tools. The tool you use is just something you call it a hyperparameter. So yeah, there's no hard and fast answers, unfortunately, for that. But it's trying them and looking at your data and finding something that works for your set of data.
Wonderful. Great. Okay, so I guess the schedule is that we move on to the panel discussion, or are there any other questions for Finn? Actually, I'm sure there must be.
We got another 5 minutes. Let's run through a few. Does Shovill work for ONT long read data? No. Shovill is only designed for short read Illumina data, I believe. However, there are some tools out there that are essentially Shovill for long read. There is, I think it was Dragonfly by the developer of, and I'll dig these up and post them in, by the developer of that Backtopia workflow. There's also Tricycler, which produces it's less of a quick tool, but produces high quality nanopore assemblies from Ryan Wick. And again, I'll post both of them in the chat. What parameters are usually altered in QC workflow, and what situations should they be changed in? Again, I know it's very frustrating when you're learning. When you're learning the bioinformatics side, we really don't like making hard and fast rules. In some data, you can even get away without doing any trimming at all, for example. But in some cases you're fine with a bit more contamination than others. It really depends on how sensitive your problem is. Some genomes are much harder to assemble than others, so they're going to be more disproportionately affected by low quality reads or contamination in your data. Is there a tool you can recommend to map plasmids and AMR genes within them? Yeah, there's a again, could be a whole workshop in itself. There are many tools to look at plasmids. One, for example, would be MobSuite. Generally, the approach most people use is you kind of separate the two problems. You detect AMR genes in your entire genome, including any mobile elements, plasmids, etc. And then you also look at what bits of your genome do seem to be mobile genetic elements, or plasmids. So MobSuite would be one tool for doing that latter case. And then you can look at your AMR genes and look at where they're mapping, where they are, to see whether, okay, this might be a mobilized piece of resistance. Yeah, I think that's going to cover. I think that'll do for a whirlwind of that. Let's say if you have any other questions, feel free to add them, especially if you have any questions later on. Also, feel free to add them to the repo that I linked. Just if you know how to do it, you can click the Issues tab, make an issue, and I'll be able to answer them for you there.